Hello and welcome to another episode of the Science on the Edge podcast. I'm Dr. Mike Darcy, and in today's talk, I'm going to be discussing how kidneys work, osmoregulation, which is way more complicated than one might think. So let's get started. Okay, so osmoregulation. So first of all, osmoregulation can be defined as homeostasis of the body's water content. Now, homeostasis just means maintenance of a steady state. We homeostatically maintain all sorts of parameters in our body, for example, our temperature, our blood's pH, sugar concentrations in our blood, and also salt and water, which is osmoregulation. That final one. So osmoregulation is the process of maintaining salt and water balance, so osmotic balance, across membranes within the body. This is controlled to protect cells by stopping too much water from entering or leaving them, a process called osmoregulation. Now, water content is controlled by water loss from a number of sources in the body. From the lungs, when we breathe out, we lose water. When we sweat, we lose water. But pretty much a primary means by which we lose water is in our urine, which is produced by our kidneys. But there's not just water that is excreted, there's also urea itself and a number of other small molecules too. And it's all under very tight regulation. So there's a constant input of water and electrolytes into the body. Excess water and electrolytes, salts, that kind of thing, sodium, potassium, chloride, and wastes such as urea are transported to the kidneys and excreted. This helps maintain the body's osmotic balance. If we don't take in enough fluid, then this results in fluid conservation by the kidneys. So if we don't take in enough water, then our kidneys need to maintain water and excrete less of it. Also, if we are overhydrated, we need to excrete more water through the kidneys. Being overhydrated is bad as well as being dehydrated. If you have too much water inside your cells compared with outside the cells, then what will happen is the cells will lose water and they will dehydrate. If, however, there's too much water outside the cell compared with inside the cell, then what will happen is water will suck into the cell and the cell will swell up. Basically, if there is an imbalance, there's a problem with the maintenance of the cell shape and structure, and it can either dehydrate or it can pop in severe cases, of course. Okay, so biological systems interact and exchange water and nutrients with the environment by way of consumption of food and water and through excretion in the form of sweat, urine, and feces. Without a mechanism to regulate osmotic pressure, or when a disease damages this mechanism, there's a tendency to accumulate toxic waste and water, which can have really severe consequences. So controlling water levels. So osmoregulation is the process by which organisms regulate the water content of the body. As mammals, we need to ensure the volume of blood plasma, so the liquid that the cells float around in the blood, and concentration of dissolved substances, salt, sugar, that kind of thing, in the blood and tissue fluid stay relatively constant. Osmoreceptors in the hypothalamus of the brain, just beneath the brain, monitor the water potential of the blood, so the osmotic balance of the blood. This varies the amount of antidiuretic hormone, ADH, which is released into the bloodstream. The kidneys respond to a change in ADH concentration by adjusting the volume and concentration of the urine. So basically, the hypothalamus does a lot of things in the brain. It's sort of a link between the nervous system and the hormonal system. And it controls all sorts of hormones in the body, as well as sensations such as thirst and hunger. It primarily controls the hormones in the body by controlling another gland called the pituitary gland, which hangs just beneath it. The pituitary gland is about the size of a bean and secretes a total of 9 hormones under the influence of the hypothalamus. An antidiuretic hormone is just one of them. It's actually secreted from the posterior pituitary gland, the back of the pituitary gland. So let's explain osmoregulation. So here in pink, we have the hypothalamus right here. And below it, we have the pituitary gland hanging just below it. Hypothalamus controls the pituitary gland. Now, one of the things the hypothalamus does is it monitors the water potential of the blood. So if the normal water potential decreases, so if we become dehydrated, this is detected by osmoreceptors in the hypothalamus. These osmoreceptors, specialized cells that detect how much water is in the blood, then fire. Basically, if we are dehydrated, then the hypothalamus makes the decision that we must conserve water. So what it does is it tells the pituitary gland to release more antidiuretic hormone. Antidiuretic hormone goes to the kidneys and increases the permeability of the collecting ducts within the kidney. This allows more water to be retained and less water to basically go from the kidney to the bladder and be urinated away. So the urine becomes more concentrated, so less dilute, and this restores normal water potential. If, however, there's an increase in water potential, meaning you are overhydrated, this is bad too. Once again, this is detected by osmoreceptors in the hypothalamus, and a decision is made that water must be expelled. Signal is then sent from the hypothalamus to the pituitary gland, telling it to release less antidiuretic hormone, which causes a decrease in permeability of the collecting ducts, meaning more of the water that goes to the kidneys from the blood is allowed to go through the kidney to the bladder and is urinated away, making the urine more dilute, more watery, and restoring normal water potential. So this is known as a negative feedback loop, and it's how we homeostatically maintain the water and salt concentrations within our body. So here is a kidney. Now, as you can see, we have here the renal artery going in. We have one going to each kidney. This is in red. And then the renal artery branches off into many arterioles. And each arteriole connects to a nephron. Now, this one is obviously zoomed in. A kidney actually contains about 1 million nephrons, but they are this long. They go from the cortex of the kidney, the outer part, right to the pelvis in the middle. And this is the shape of a nephron. There's just about 1 million of them. Blood goes to a nephron where some filtration takes place and waste products are removed by entering the nephron. They then go to the pelvis of the kidney and then to the ureter, which goes to the bladder. The 2 ureters, one from each kidney, go to the bladder, and then the urethra takes the waste out of the body as urine. Now, this nephron is actually way more complicated than it looks. It's not just that liquid goes in and waste products and they leave. There's a constant movement in and out of this nephron, meaning that we retain what we want to retain and only excrete what we want to excrete. So the kidney's role, maintenance of salt and water balance and the elimination of nitrogenous and other metabolic waste. Nitrogenous waste, by the way, just means the breakdown products of protein. You see, we can store carbohydrates in our body as glycogen in our muscles and our liver, and we can store fat in our adipose cells. We cannot store protein. So protein, any protein that isn't used, is then broken down into nitrogenous waste products, urine, urea, and other metabolic wastes. Blood is then filtered into the renal tubule, which is the nephron I just showed you, by ultrafiltration under the force of the blood pressure. The nephron is the basic functional unit which produces urine in vertebrate kidneys. It's comprised of the following components: Bowman's capsule, glomerulus, proximal convoluted tubule, PCT, loop of Henle, distal convoluted tubule, DCT, and the collecting duct. And I'll explain what each of these do very shortly. So here is a nephron and its associated capillaries. Remember, there's about 1 million of them in each kidney. Now, first, I'll just sort of list what's going on here. We'll probably keep going back to this diagram as we move along, because it's hard to kind of just sort of understand what's going on just from reading the text. You need a visual aid. So here is a branch of the renal artery. It goes in here to the Bowman's capsule, the beginning part of this structure, the nephron. This is where many products leave the blood supply and enter the nephron. So this curly bit here is the glomerulus. It's a very thin blood vessel. Now, as you can see, when it leaves, so it goes in as the afferent arteriole. Afferent just means going in. Efferent means exiting. As you can see, the blood goes in and leaves, but the afferent arteriole is wider than the efferent arteriole, which means pressure builds up inside. So much pressure that small molecules, including water, salts, amino acids, vitamins, sugar, a bunch of things, are pushed through the membrane of the glomerulus into this first structure, the Bowman's capsule. They then go down into the first part of the nephron, which is the proximal convoluted tubule, this curly bit, then the descending arm of the loop of Henle, then up the ascending arm of the loop of Henle to the distal convoluted tubule, this curly bit here, the collecting duct, and then the collecting ducts of each nephron join at the pelvis of the kidney where the urine is excreted. So the nephron, about 1 million in each kidney, and they're associated with lots of blood capillaries. Now, one thing I should say actually is I've just talked about the nephron here, but as you can see, surrounding the nephron are all these blood vessels. And this is where exchange occurs because the nephron is permeable to certain substances, partly controlled by hormones, such as aldosterone and antidiuretic hormone. So certain molecules can leave the nephron where they go back into these capillaries, then into a branch of a renal vein, and then the renal vein itself, and go back to the body. So anything that's retained just goes back to the body. So each nephron starts in the cortex, the outer part of the kidney. Capillaries form a knot called the glomerulus, surrounded by the cup-shaped Bowman's capsule. Fluid from the blood is filtered into the capsule by the process of ultrafiltration. The capsule leads into the nephron, which consists of the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct. As fluid passes along the nephron, its composition is altered by a process known as selective reabsorption. Substances are reabsorbed back into the tissue fluid, then blood capillaries surrounding the nephron. The final product in the collecting duct is urine, which passes into the pelvis through the ureter to the bladder. So now let's look at the glomerulus and the Bowman's capsule. So we've zoomed right in here. So this is the glomerulus. This is the cup-shaped Bowman's capsule. Now, blood cells, red blood cells, white blood cells, etc., are too big to pass out through this filtration process. Only very small molecules can leave. So let's explain how this works. So blood enters the glomerulus through the afferent arteriole. It's wider than the efferent arteriole that carries the blood away. Because more blood is entering the glomerulus than can leave at any one time, pressure builds up. The pressure forces water and small molecules out of the blood through small pores in the endothelial walls. That's the cells that line the capillary, the glomerulus. These pores are sometimes called fenestrations, little holes. The filtrate meets the basement membrane, so the membrane surrounding the glomerulus. This is made of a mesh of collagen fibers and glycoproteins, which just means proteins with sugar on them. It acts as a filter, kind of like a net, and will only let molecules smaller than about 69000 relative molecular mass through. That just means only very small molecules, not things like blood cells. So big proteins and blood cells are too large to pass through the basement membrane and therefore stay in the blood. If you see blood in the urine, by the way, or even proteins, you might not see blood cells, but you might see proteins. If you see those in the urine, then that means there is damage to the kidney, quite often caused by high blood pressure. The kidneys are very sensitive to high blood pressure, and either blood or more often proteins in the urine is a sign of kidney damage. As most of the liquid content of the blood has been lost, but it still has large proteins in it, this greatly reduces the water potential of the blood. So it makes it more negative. For want of a better word, more dehydrated. The epithelial cells of a Bowman's capsule, so that's the cells that line the inner part of the Bowman's capsule, the cells that are in contact with the glomerulus, are called podocytes. This is from Greek. Basically means foot, because they have little feet on them. So podocytes have large finger-like projections, the feet, known as major processes or pedicles. The pedicles ensure that there are gaps between cells for the filtrate to pass through and to drain into the inside of the proximal convoluted tubule here. So this process of ultrafiltration removes a lot of products from the blood. So by the time you get to the proximal convoluted tubule, lots of small molecules have left the blood. Water, amino acids, which are the building blocks of proteins, glucose, urea, inorganic ions such as sodium chloride and potassium. Can you see a problem with the substances that have been removed from the blood? We need some of them. Yeah, we want to get rid of the urea. We might want to get rid of some of the water and salts, but we also want to retain some of these things. We certainly want to retain glucose and vitamins for that matter. Water-soluble vitamins also leave the blood and go into the proximal convoluted tubule. So here's the nephron again. So ultrafiltration happens at the Bowman's capsule, but many useful substances have been removed from the blood. They need to be reabsorbed. And in the proximal convoluted tubule, most of them are reabsorbed. 85% of the filtrate, in fact, is reabsorbed back into the blood in this region, in the proximal convoluted tubule, right here. From the proximal convoluted tubule, the filtrate moves into the descending arm of the loop of Henle, then the ascending arm of the loop of Henle. The main role of this section is to concentrate salt in the tissue surrounding it. So I'm going to keep jumping back here. So as you see, here at the proximal convoluted tubule, 85% of the filtrate has now gone back into the blood, back to the body. The rest goes into the descending arm of the loop of Henle, then the ascending arm of the loop of Henle. So the main role of the loop of Henle is to concentrate salt in the tissues surrounding it, so in the kidney itself. So it begins in the cortex, the loop of Henle from the proximal convoluted tubule, extends down into the medulla, the middle of the kidney, and back up to the cortex. So the loop of Henle goes down, then up again. The descending and ascending limbs have different properties. The descending limb is permeable to water and less permeable to salt, so water can leave by osmosis. So basically, in this part, the descending arm of the loop of Henle, salt can't really leave, water can. So water can come out and go back to the blood. Now, in the ascending arm, this is impermeable to water, but actively transports salt. So here, water cannot leave, but salt is actually pumped out. to the surrounding tissue. This makes the kidney salty, basically, which actually helps water to leave the nephron from other parts of the nephron. Because if it's salty and you open a gate to let water go out through the nephron, it will go back into the blood. It will follow the salt by osmosis. The filtrate then gets to the distal convoluted tubule where further reabsorption takes place. So this is the distal convoluted tubule. More reabsorption takes place here. And then it goes to the collecting duct, where it is taken to the ureter, then into the bladder. Okay, if any of this is confusing, just rewind a little bit and take another look. Now, hormones play a role in all of this. So the hormone antidiuretic hormone, little spelling error there, sorry about that. So antidiuretic hormone, ADH, known as vasopressin 2, regulates water balance by controlling how much water is reabsorbed in the kidneys. ADH increases water reabsorption, which reduces urine output and helps maintain blood volume and blood pressure. Another hormone, aldosterone, controls the balance of sodium and potassium in the body by promoting the reabsorption of sodium in the kidneys. So reabsorption of salt, while excreting potassium. This hormone is regulated by the renin-angiotensin-aldosterone system, RAS, which is activated when blood pressure is low. Blood pressure is too low, you need more salt in your blood so that water will reabsorb too, and the blood pressure will increase. The hormone atrial natriuretic peptide, AMP, promotes the excretion of sodium and water in the kidneys, thus reducing blood volume and blood pressure. AMP is released in response to high blood pressure and acts to counterbalance the effects of aldosterone. So if blood pressure is too low, you basically want to stimulate aldosterone to get water and salt back in the body and increase blood pressure, which basically means blood volume as much as anything else. If blood pressure is too high, then AMP can counteract that and lower blood pressure, allow you to get rid of more salt water from the body. Overall, hormones play a critical role in regulating water and salt balance and blood pressure in the body, and any imbalance can result in serious health problems. So now let's zoom in on how some of this works. So what we're looking at here, this is the inside of the proximal convoluted tubule. So the inside of the nephron at the proximal convoluted tubule. This is a layer of cells that lines the tubule. So this is the tubule, basically. Inside and outside. So surrounding the tubule are these blood vessels. Now, this is a little bit molecular, but we're going to explain here just how we reabsorb the filtrate back into the blood. This requires energy in the form of ATP. ATP is the energy currency of the cell. We spend ATP to power things. That's why we breathe oxygen, why we eat food, to make ATP, and then we spend ATP to power things, to operate our muscles, pumps within our cell membranes, all sorts of things. It's made in our cells primarily by the mitochondria. So, 1. Sodium-potassium pumps in the basement membrane of the proximal convoluted tubule cells actively transport sodium out of the cells into the tissue fluid. So this is a pump. It spins around, it grabs sodium from the inside of the cell and pumps it out to the surrounding tissue fluid. This reduces the sodium concentration in the cell because it's being pumped out. There's now no sodium inside the cell. Mitochondria provide the ATP required to operate the pump. Now, this creates a diffusion gradient for the movement of sodium ions from the filtrate through cold transporter proteins in the PCT. So basically, any salt that's in the urine here in the proximal convoluted tubule wants to come into the cell because there's no sodium inside, so it wants to fall down its concentration gradient like water going down a waterfall. But to do that, it has to go through this cold transporter. It has to go through a cold transporter. Now, this cold transporter is like a spinning door in a building. There's a channel for sodium. There's also a channel for glucose and amino acids. So as the sodium falls down that waterfall, so to speak, it spins around this cold transporter like a water wheel. It gets into one slot, it spins around, and the other slot grabs some glucose or amino acids and also pumps that into the cell, so gets it out of the filtrate. So this is an example of facilitated diffusion, as the concentration of glucose and amino acids is now high in the PCT cell. This reduces the water potential of the cell and creates a diffusion gradient for movement of water from the filtrate into the PCT cell. So the water follows the salt, so to speak. As the concentration of glucose and amino acids is now high inside the cell, this creates a diffusion gradient for the movement from the cell into the tissue fluid surrounding it, and then into the blood capillaries and back into the circulation. As this has now reduced the water potential of the blood, water will also move from the PCT cell into the blood. This is showing you basically what happens in the next parts of the nephron. So here in the loop of Henle, the distal convoluted tubule, and the collecting duct. So you can see that water can leave here, salt leaves here, water leaves here. So sodium and chloride ions are actively transported out of the top of the ascending limb of the loop of Henle into the surrounding tissue fluid in the medulla, the middle of the kidney. This builds up a salt gradient in the medulla with high salt concentrations near the pelvis and lower concentrations near the cortex. As the walls of the ascending limb are impermeable to water, water cannot follow by osmosis. The loss of salts but conservation of water increases the water potential of the filtrate, which makes it quite dilute. The descending limb is very permeable to water. As the filtrate moves down the descending limb, it comes into contact with the increasingly salty surroundings of the medulla. Water therefore leaves the filtrate by osmosis and enters the tissue fluid in the medulla. The result is that at the bottom of the descending limb, you end up with a very concentrated filtrate with a low water potential. There is some diffusion of salts from the medulla back into the descending limb. This allows a maximum diffusion gradient for salts into the medulla at the base of the ascending limb. The water entering the tissue fluid in the medulla would dilute the salts there if it were allowed to accumulate. It therefore diffuses directly into the capillaries of the vasa recta, which are the capillaries surrounding the nephron, and is carried immediately away. So the arrangement of a loop of Henle is what we call a hairpin countercurrent multiplier. The overall effect of this arrangement is to increase the efficiency of salt transfer from the ascending to the descending limb, which consequently causes the buildup of the salt gradient in the medulla. As the filtrate moves into the distal convoluted tubule, active transport is used to adjust concentrations of salts in it. Then the filtrate flows into the collecting duct. The filtrate is dilute at the top of the collecting duct. It has a high water potential. The surroundings of the medulla are salty, so water leaves the filtrate and goes into the tissue fluid in the medulla by osmosis. This reduces the water potential of the filtrate, but as it is now coming into contact with higher salt concentrations in its surroundings, there is still a good gradient for the movement of water into the medulla by osmosis. So to summarize this, blood enters each kidney from the aorta, that's just the main blood vessel leaving the heart, via the renal arteries. It is distributed in smaller vessels until it reaches each nephron. These are these arterioles. Within the nephron, the blood comes into intimate contact with the waste collecting tubules in a structure called the glomerulus. Water and many solutes present in the blood, including ions of sodium, calcium, magnesium, and others, as well as wastes and valuable substances such as amino acids, glucose, vitamins, etc., leave the blood and enter the nephron. Now, some of these are needed, so they need to be reabsorbed. This requires ATP. Some wastes, including ions and some drugs remaining in the blood, diffuse out of the capillaries into the interstitial fluid and are taken up by the tubule cells. These wastes are then actively secreted into the tubules. The blood then collects in larger and larger vessels and leaves the kidney in the renal vein. The renal vein joins the inferior vena cava and basically goes back into circulation. The amounts of water and ions reabsorbed into the circulatory system are carefully regulated by means of hormones such as aldosterone and ADH. The waste is collected in larger tubules and then leaves the kidney in the ureter, which leads to the bladder where urine, the combination of waste materials and water, is stored. I should point out at this point that it's essential that you get rid of waste products, but especially things like the breakdown products of protein, the nitrogenous wastes, because if they accumulate, they're very toxic to the body. Now, blood pressure needs to be maintained partially by the kidneys. Now, changes in blood pressure are routinely made in order to direct appropriate amounts of oxygen and nutrients to different parts of the body. When you exercise, you need to increase blood pressure. When you sleep, you need to reduce it. Now, blood pressure can be adjusted by producing changes in certain variables, cardiac output and stroke volume. So if you increase cardiac output, the amount of blood that leaves the heart per minute, by partly increasing the number of beats per minute, but also the stroke volume, which is the pressure that the heart beats at, how much blood leaves the heart in one beat. So that's partly how you can do this. Resistance to blood flow in the blood vessels is most often altered by changing the diameter of the vessels. So vasodilation widens them, lowers blood pressure. Vasoconstriction, constricting the blood vessels, increases it. Also changes in the viscosity of the blood, so how much salt and water is in there. Now, weight gain has an effect as well, because if you have a very large body, obviously your blood vessels are longer to get blood around that body, which means there's more resistance in there to blood flow, and that can obviously affect blood pressure too. Now, there are parts of the brain that control all of this. So the cardiovascular center, which is in the medulla oblongata of the brain, in the brainstem, provides a rapid neural mechanism for the regulation of blood pressure by managing cardiac output or by adjusting blood vessel diameter. Now, the cardiovascular center receives information from different receptors in the body. For example, baroreceptors. Baroreceptors detect blood pressure. Think of them as kind of sensory neurons lining the arteries that will fire when the artery stretches, when it gets wider, and then send a signal to the brain, telling the cardiovascular center that blood pressure is high. Now, these are located in the carotid artery and in the aortic arch. So carotid artery, the artery going to the brain, aortic arch, artery leaving the heart. They monitor these changes, medulla is informed and makes a decision. Now, there's chemoreceptors as well. These, again, are receptors in the blood vessels. They're found in the carotid bodies, near the carotid sinus, and in the aortic arch again, the heart and brain being very important regions for maintaining blood pressure. And they monitor carbon dioxide levels and and oxygen levels. Obviously, if CO2 gets too high, then usually when you're exercising, for example, then this means you need to expel it and to get more oxygen in, because it's usually a byproduct of oxygen consumption. So these chemoreceptors fire and tell the brain, we need to increase blood pressure and heart rate so that we can get the CO2 out and oxygen in. And then there's higher brain regions too, in the cerebral cortex, the outer part of the brain. There's also the hypothalamus and other parts of the brain, which also are involved in maintaining blood pressure, for example, in response to stress. Now, the kidneys provide the hormonal mechanism for all of this. So ADH, produced by the hypothalamus, released by the pituitary gland, raises blood pressure by stimulating the kidneys to retain water. Aldosterone also directly affects the heart and blood vessels. The aldosterone hormone is a hormone produced by the adrenal gland, the adrenal cortex, the outer part of the adrenal gland. It's the middle that produces adrenaline, by the way, the medulla. The hormone acts mainly in the functional unit of the kidneys to aid in the conservation of sodium, secretion of potassium, water retention, and to stabilize blood pressure. Aldosterone also causes water to be reabsorbed along with sodium. This increases blood volume and therefore blood pressure. Thus, it indirectly regulates blood levels of electrolytes, sodium, potassium, hydrogen, and helps to maintain the pH of the blood. Okay, that was a briefish summary of how the kidneys work. Thank you very much for watching. If you want to know more about me, then you can follow me on Twitter. My handle is @MarkOfTheD. So thank you very much for watching. I look forward to speaking with you all again soon, but goodbye for now.